class then. So let me get the PowerPoint up here. All right, so today we're going to finish talking about kind of the tail end of chapter 1 in terms of talking about the research methods that are pretty commonly used in cognitive psychology. And so as you can imagine, especially since this is a class that has a lab component attached to it, lab studies that use an experimental methodology to them. So that is to refresh your memory. So experiments are research studies that involve the manipulation of one or more variables. And that's in contrast to research that can be done via pure observation. So just like it sounds, you're sitting and observing somebody's actions or the outcomes of a certain phenomenon. And observation is not experimental. That is, it does not involve any manipulation. So lab stories, well, maybe, but lab studies are really core to cognitive psychology research. And we're going to talk about research methods. It's going to hopefully feel like a little bit of a refresher from your experimental psych class. And we're obviously covering this because, you know, the end of the text, or the end of the chapter for this week talks about research methods, but I'm also hoping too that this lecture will dovetail kind of nicely with the beginning of the design your own study lab that we had this week. I know that you all were brainstorming really hard. I am so sorry. My cat has the zoomies right now in this moment, so that's perfect. I know that, yeah, all of you were really coming up with some really interesting ideas. Some of you had even started thinking about, okay, well, how will we go about investigating this? And some of you hadn't, and that is totally fine. But I'm hoping that this portion of the lecture can also start to help you as you do start to go from the conceptual brainstorming part to the more logistical side of, okay, well, how are we going to run the study? And like I said, this is just going to feel like a little bit of review from experimental psych. But as you might remember, we try to conduct research in the lab when we can, because it gives us the ability to control as many variables. So meaning things that are in the environment, such as the room temperature, but also things that our research participants encounter, such as the set of pictures that are shown to them in a study, for example. And the reason why we like the lab is because the more control that we have over variables as researchers, the more clearly we can study cause and effect. So rather than sitting and observing out in the real world, we can set up a simulation in the lab to try to be as similar to the real world as possible in order to say, okay, we do have some evidence that A causes B, or A does not cause B. And so in talking about variables, so there are 3 categories of variables. So there are independent variables, there are dependent variables, and then there are what we would call extraneous variables. And these are the variables that we want to control for. And I'll talk about each of these. So as a reminder, our independent variables are the things that we are manipulating in our experiment. So for example, in the context of cognitive psychology, this might be the presentation speed of a list of words read off to participants. You know, perhaps you're reading 1 word a second versus 3 words a second, or 5 words a second, you know, something like that. It can also be something like the emotional content of video clips that you are showing to participants. You know, are you showing them a clip from a horror movie trying to induce fear, things like that. Okay, so the independent variables, the IVs, those are the factors of the study that we as the researchers are thoughtfully and intentionally manipulating. In comparison, the dependent variables are the things that we're measuring. So for example, we might be measuring people's reaction time to words that are flashed on the screen. If you're familiar with the IAT, the implicit association task, you're measuring, well, how quickly does somebody respond to a word? We also could be measuring something like the number of items that are completed on a task. So it's not in the context of an experiment, but technically speaking, if you wanted to, you could say that a test score is a dependent variable. So it's something that we are measuring. And the dependent variable is really important because not only is it measuring, oh, okay, you know, this particular number, right, of like, oh, reaction time in milliseconds, but it's how we're quantifying or putting numbers to somebody's performance. And I know that the terms independent variable and dependent variable, it's hard to kind of keep them straight. You know, it's like, okay, well, why do you both have to have very similar names? But the dependent variable is called dependent because it is assumed that that will change based on the impact of the independent variable. So there's this assumed contingency or connection between the dependent variable, the thing that you're measuring, and the independent variable, the thing that you're manipulating. So let's take an example. So let's say that we were wanting to examine the impact of caffeine intake on people's reaction times. So in this case, our independent variable that we are going to manipulate, the thing that we have control over, is going to be caffeine intake. And let's just say this is in the form of just a basic black cup of coffee, 12 ounces, whatever. So let's pretend that in this experiment, we are going to have 3 groups. Let's say we have one group doesn't get any coffee to drink before the experiment. If you like coffee, you might be excited to be in, not to be in that group. If you don't like coffee, you might be like, oh, great. Maybe there's another group that is asked to drink 2 cups of coffee before starting the experiment. Maybe there's another group that's then asked to drink 4 cups of coffee, which I really hope I wouldn't be put in that group because I have enough heart palpitations with just like 1 large cup of coffee, which I always forget. And then I get like a venti and then I'm like, why do I feel like I'm dying? Caffeine. So in this caffeine study example, our participants' reaction time performance. So that is the dependent variable, right? How quickly they are responding to something. It's assumed that that response time depends on how much caffeine the participants in our study have consumed. And so, possibly we might think, maybe more caffeine leads to or directly causes faster reaction time. Or you might think, oh, well, it could go the other way. More caffeine might make you so jittery that it actually directly causes slower reaction time. And then the third category of variables that we want to talk about and think about are those that are what we call extraneous or outside of your independent variable and dependent variable that you will want to control for. So taking our caffeine example, a variable that you would want to control for is the amount of sleep that your participants got the night before they came into the experiment. Can anybody tell me why we would want to know about this variable?
Because sleep could make people like less reactive or more reactive, and then it could be an extra variable that's needs to be controlled because then it's not just the caffeine that's affecting them, it's their sleep.
Right, exactly. Well said. So the amount of sleep could be a variable that impacts our dependent variable. So this makes it more difficult for us to say conclusively that our experiment was looking directly at the impact of caffeine on response time. Sleep could also be something accounting for that. So that's something that we need to control for. And in this sort of instance, it could be just as simple as, asking participants at the end of the study, hey, by the way, how many hours of sleep did you get last night? Did you wake up feeling rested this morning, or did you wake up feeling groggy? Um, so it doesn't have to be anything elaborate per se, but being aware of what kind of variables could also be leaking into your study and impacting the dependent variable in ways that you don't want to is really important as well as we're doing research. Okay, and then, so going back to the idea of dependent variables, so there tend to be two main types that are involved in cognitive psychology research. So the first one that you'll see, it's pretty popular, is measuring participants' accuracy. So that could be in the form of the number of items that are correct, or perhaps the percentage of items that are correct. Or you could also decide to measure errors, so the number or percentage that's incorrect. And then, like we talked about in the caffeine study example, reaction time is another really popular dependent variable. And so this is the speed at which your participants might click a button to respond to a question, or the amount of time it might take for them to sort an item into a category you've given them, or the amount of time it takes for them to verbally produce a response to your question, and et cetera. And then, so regarding independent variables, there are also some common kinds that tend to be more popular in cognitive psychology. And so, for example, we have characteristics of the situation. So these could be elements like the lighting that's in the room, the temperature of the room, the noise level. It could also be the nature of the stimuli that you are having participants engage with. So this could be maybe whether there's visual stimuli, like showing them a picture, versus verbal, like you reading off a list to them, different ways in which stimuli are presented. There's also specific characteristics of the task, or like the logistics of the task. So this could be related to questions about, okay, well, were participants given a time limit in order to respond, or were they given as much time to respond as they wanted? Did they have to freely recall information? Let's say you read them a list and then 20 minutes later you say, okay, tell me all the words that you remember on that list. Or do they just have to recognize what was presented to them earlier, either reading off a list and saying, hey, tell me which words you remember, or circling words, etc. And then we also have the characteristics of the participants themselves. And so this could involve factors such as their age, their experience that might be related to that particular task that you had them do, maybe previous scores, level of education. If you're having somebody who has a certain level of education and you're comparing it to another group of people. Gender, of course, is a big one, as well as ethnicity. So there are a lot of different variables that we can try to manipulate or focus on to see how they might then impact our dependent variable. And so in addition to studying cognitive processes based on, our participants' behavior that they demonstrate in the lab or their performance, we can also use other methods to study human cognition as well. And so another way to do this is through neurological research. And so this type of research looks at the relationship between our cognitive abilities, so the level of analysis is on what we would call the mind, and also the brain. So this brain and mind or brain behavior connection. And the brain is sort of being defined as, okay, well, these, cerebral events that are taking place, and then the associated structures within the brain. And so there are a variety of neurological techniques that can be used in cognitive psychology. One technique is a post-mortem study of the brain. And so this is a physical examination of brain tissue after someone has died. And it frequently involves the use of neuroimaging, so using something like a CAT scan, which we'll talk about more in depth later in the lecture. It also very frequently involves dissection, so cutting the brain into slices in order to then study the appearance of particular brain structures microscopically. So this could be focusing on the hippocampus, which plays a big part in memory. And you could also be even looking at a more microscopic level than the structure. So you might be looking at, let's say, groups or networks of cells. And the reason why we want to do post-mortem studies is that there are several conditions or diseases that cannot be verified with like 100% certainty until the person has passed away, unfortunately. Does anybody know of any conditions or disorders in which this is true?
Is Alzheimer's one of them?
Yes, Chase, that's correct. Alzheimer's is one of them. So we have Alzheimer's as one of them. And so Alzheimer's disease, what happens in the brain is there's this ongoing deterioration of cellular components. And so kind of these two, the two hallmarks that we talk about on imaging and brain dissection when we talk about Alzheimer's is what are called beta amyloid plaques, or just amyloid plaques, which in this sort of more cartoony depiction are these brown spots here. And then also these neurofibrillary tangles, which in this cartoony depiction are these purple structures that have these extensions that they're really fine, and they are very easily tangled up on one another. And this picture over here to the right is actually from a microscope examination of a postmortem brain in a person who was suspected of having Alzheimer's and had been diagnosed as having Alzheimer's. But again, without this final piece, it was like, okay, well, we're pretty sure by giving you this diagnosis, we're pretty sure that this is what's going on, but we can't actually know until you've passed away, unfortunately. But here on this microscope slide, you can see this arrow here, these parts that are stained pink, these are the amyloid plaques. And then these parts that are black and are larger in size, these are the neurofibrillary tangles. And so this is one such condition that we do have a lot of evidence for clinically. So we can see it in people's lives, maybe their decline in social functioning or occupational functioning, but we can also see it on cognitive testing as well. However, like I said, we can't confirm it and say, yes, this was 100% Alzheimer's until there's a post-mortem examination. And another condition that can't be fully diagnosed until death is CTE. And CTE, this stands for chronic traumatic encephalopathy. So you can see why CTE is the go-to, because it's a mouthful. And this might sound familiar to you. It's been in the news over the past couple of years. And research has shown that this condition is associated with a history of a large amount of serious head injuries. And so many CTE cases have been seen in professional athletes who play sports, heavy contact sports that result in a lot of head injuries or a higher likelihood of head injuries, such as football, hockey, boxing. And these repeated injuries lead to this really global or overarching loss of brain tissue. It's not focused in just one area, but it really impacts the entire brain. And this can only be verified upon autopsy. This GIF is actually from a New York Times study. And you can see here they kind of have it broken out into categories of the position that these football players had. And you can see the brown staining is really showing areas of pretty advanced deterioration and cell death. And again, these are all from brain slices of these football players who had passed away and had shown characteristic behavioral signs of CTE when they were alive. Okay, and so in addition to post-mortem studies, we are also able to study living humans to look at the relationship between cognition and the brain. And we can do this by examining the outcomes in people who have suffered some kind of brain damage. So a famous example is Phineas Gage. Does anybody recognize that name?
Yeah.
Okay.
He like
can you
mine, right? And then he like got a pole stuck through his head.
Yep, exactly. Yeah. So this happened back in the mid 1800s. And so he was part of a crew that was building a railroad. And so what they had to do is they had to just use dynamite to blow out the road so that they could then set in the railroad tracks. And so he was using what's called a tamping rod. It's this big, I think it's a couple of inches in diameter, and it weighed about 40 pounds, or sorry, 14 pounds. And he didn't realize that there was dynamite that was already laid down there. And so he hit this iron down, it exploded up into his the left part of his face, and it went through past his eye socket, and it actually ripped through his brain and shot out of his head and landed a couple feet away. And so because of this, he was obviously blind in his left eye, but amazingly, he didn't lose consciousness. He was still able to talk coherently. He amazingly didn't bleed to death. And so people were like, wow, okay, this is crazy. You should have died, but you didn't. Cool. All right. Well, once you heal up, everything should go back to normal. But unfortunately, after this, his friends and family really described him as someone completely different. So he had difficulty sticking to plans that he had made, being organized. He behaviorally, he was really rude. He swore up a storm. And this was in complete opposition to who he was before. He had been very responsible, very kind, very friendly, and now he seemed to be the exact opposite. And so this was one of the first instances that really showed us that, okay, the things that happen to the brain can actually impact the way that we think and go about our lives. And so some more examples of what we can look at to study the relationship between cognition and brain damage are conditions like strokes, traumatic brain injuries, the possible impact of tumors that might be growing on the brain and pushing into a certain area that is then disrupting the function of those underlying structures. Sometimes there is instances of neurosurgeries that have to take place in order to remove some sort of harmful brain tissue. You know, there might be a cancerous tumor as well. And so this is another way in which we can study the brain and our behavior. Another source of research are studies carried out with animals. And this isn't done because we assume that animals behave the same as humans, like in the case of behaviorism, but this is done for ethical reasons. So, for example, if we were interested in what happens cognitively when the hippocampus is damaged, for example, we would have to depend on cases that have happened in people that have been the result of just, sort of natural happenstance. So maybe there was some sort of injury, right? Like in the case of Phineas Gage, if we were interested in, what happens to the frontal lobe when it's damaged, we could look at somebody like Phineas Gage. Or if somebody had to, let's say if they had to undergo a necessary brain surgery, right? Maybe there was a tumor, or they're having very severe seizures that are originating from a certain part of the brain, and it's not responding to anything else, then, maybe that brain tissue needs to be removed as a last resort. And so the thing is, we can't recruit people for a study in which, say, half of them would have, one, either their left or right or both hippocampus destroyed, and the other half doesn't, in order to kind of see what happens. We can't say, okay, well, we're going to manipulate this and you lose part of your brain and then you don't. That's thankfully unethical and illegal. And so because of this, animals with similar brain makeup and structures to humans, such as rats, are used. And it is ethical to use an animal for research purposes when the procedure can't ethically be carried out on a human, such as this example of hippocampus destruction. But again, that's pretty much the only reason why cognitive psychologists would turn to animal studies. And then, of course, another great way to study cognition and the brain is through neuroimaging. And so there are several go-to neuroimaging techniques. And so one of them is called CT or CAT scans. It's interchangeable. They're the same sort of imaging technique. CT stands for computerized tomography. And I'm going to go ahead and jump ahead and just show you. So this is an example of a CT on the left side of the screen. And so with CT scans, they involve taking multiple X-rays and then compiling them into one image in order to sort of show slices of the body. So like you can see here, this is kind of a slice that has been photographed in this CT. Sorry, Chase, did you have a question?
Yeah. So for Alzheimer's and only being able to be diagnosed posthumously, why would there not be able to be like a biopsy done or an MRI? Like, why wouldn't those work? Like, why would you have to wait until you could actually like take the brain out?
Yeah, that's a great question. And there are some techniques that are allowing us to be more precise. So for example, there is a kind of imaging, and I don't remember what it all stands for, but it's called an FDG PET scan. And it allows us to then get a sense of the amount of the beta amyloid plaques and the neurofibrillary tangles that are in the brain. But again, without being able to isolate under a microscope those plaques and tangles, as we know that those really are the hallmark signs, it is difficult for us to say with 100% certainty. We have like a lot of clinical evidence that we can point to. With Alzheimer's disease, there tends to be a pretty specific pattern of cognitive degeneration. So we can have evidence for that. We can have, like I said, evidence from some of these imaging where there are certain structures of the brain that we know, yeah, this tends to be impacted by Alzheimer's disease. But it's not until you get to a post-mortem examination that you can say that for sure. Because Alzheimer's it's a subtype of dementia, and sometimes other types of dementia, they might look like Alzheimer's, or maybe at the beginning of Alzheimer's, for example, it might look like another type of dementia. And so at this point, the post-mortem study looking for those plaques and tangles is really the only way that we can say for certainty. Unfortunately, the thing with the brain is that it's really hard to biopsy without causing damage. If you have somebody who's already having cognitive decline, it's like, oh, we really don't want to jeopardize that. And, they're usually older. And so then there are complications that go into that as well. So yeah, right now we don't have a better way, unfortunately, of confirming that with certainty until the person has passed away. But Lily, did you have a question?
Yeah, just kind of going off what Chase said about like the postmortem autopsies. I don't know. Is it expensive or would like you be donating to science? So like, because I feel like some of it might be like valuable information, like it would ease people, like if like I know for the CTE, like some families like want to know like if that was the cause. So do you have to pay for that?
It's a good question. And typically in just sort of the general public, because of things like costs, which are prohibited, but also, as you can imagine, families might not be too keen on the idea of, you cutting into your deceased loved one's brain. It really is more based on a donation sort of, in that people who are suspected, quote unquote, but really diagnosed with having Alzheimer's, then opt to donate their brains to science in order to get that. And so because it is, emotionally difficult and financially difficult, it's not common. So it's not like every single person who is given a Alzheimer's diagnosis in life is then followed up on. But at least at the level of really extensive research, that is the way in which the researcher are able to say, yes, this person was in our study. We had put them in the Alzheimer's group, and we had followed their progression. And now at death, we are able to say positively, yes, they were in the Alzheimer's group. Their pattern of cognitive decline was the result of this Alzheimer's process. Yeah, great question. If you can't tell, I'm a little bit of a brain nerd, so I like being able to talk about these things. It's fun. Okay, so going back to CT scans. So these involve, like I said, taking multiple x-rays and then compiling them into one sort of image in order to show like a slice of the body. And CT scans best show bone, blood, and soft tissue damage. One of my clinical supervisors at my practicum that I'm doing at the Long Beach VA sums it up as the 3 Bs. So he calls it the 3 Bs. CTs are good for bone, blood, and bullets. It's like, well, okay, all right. And as you can see in this CT example here, so we have some blood that has spilled out from surrounding vessels that is appearing bright white here. And so this is an example of, okay, one of the 3 Bs right here. Another source of imaging is MRI, or magnetic resonance imaging. And so instead of using X-rays, MRI uses magnetic and radio waves to generate images of the body. And MRIs are considered to be better than CT scans because they provide higher resolution, more detail, and they're particularly helpful when you're looking at the nuances of the brain and the spinal cord. So I tried to get a similar-ish slice of the brain in an MRI and a CT, just so you can see the difference in clarity. You can see here the sulci and gyri, the folds and bumps of the brain so much more clearly. You can see the ventricles more clearly in the MRI versus the CT. It's a little grainier. The edges aren't as well defined. So CT is going to be kind of your basic standard image that's used, and then the MRI is kind of the next step above that. And so CTs and MRIs, they provide us with these sort of static or still images, whereas other imaging techniques that we'll talk about, such as fMRI or PET scans, these can give us information about brain activity in more real time. So I like to think of CTs and MRIs as more like just a picture, just a snapshot in time, and fMRIs and PETs being more like movie clips or GIFs, or if you're a fan of Harry Potter, you know, the like moving pictures that they have. So speaking of these different imaging types, so fMRIs and PET scans are the kinds of images that, like I said, instead of taking a picture, they're looking more at function. So they're looking more at the brain activity itself. And so fMRI, the F just stands for functional. And a functional MRI operates similarly to a regular MRI in that it does document the structure of the body or the brain. So as you can see here, this is an fMRI. You can see it's really similar to the other MRI that we looked at. It's showing you the structure, it's giving you clear designations about the various parts, but it also allows you to track brain activity based on the flow of blood and also the oxygen signals that you're getting from the brain. So for example, here we can see that these areas are more active because they are giving out a higher signal. There's more blood activity, there's a higher oxygen signal that's there, which means that the brain is consuming more oxygen in order to function. And with a PET scan, it's a similar kind of idea in that it does look at the function of the brain. PET stands for another mouthful. It's positron emission tomography. Don't know who comes up with these names, but and so this involves the use of safe radioactive tracers that are injected into the bloodstream. And this then allows doctors and researchers to see how and where the blood flow goes. So looking at this example of a PET here, you can see that, so the sort of key for reading this is that if it's red, that means it's a lot of activity. The closer it is to red, sort of the rainbow spectrum, the more activity there is in terms of brain activity, and then blue, the closer to blue or green, the less activity. So for example, here in this scan, you can see that, wow, okay, there seems to be a decent amount of activity, and we have the frontal lobe, the parietal, not sure about the temporal just based on this viewpoint. But back here, wow, we have a lot going on in the cerebellum. Whereas, you know, here in the face, right, compared to the amount of activity that's happening in the brain, yeah, there's really not a lot that's going on in your sinuses relative to the power drain that's going on in your brain. So any questions about imaging, post-mortem brain damage studies, animal studies, anything like that? Okay, we'll go ahead and we will move on to talking about other research methods as well. So another popular one is something called a case study. And the case study involves research that's focused on just one individual. So our N, or our sample size, is just one in this instance. And so like we talked about, Phineas Gage, he's an example of a case study. And another famous case study in cognitive psychology is someone called HM. Has anyone heard of HM before? If not, that's okay. That's why you're taking a cognitive psych class. So HM was a gentleman, and this happened in like the early 1950s, and he had very severe seizures for about 10 years. He was 27. And he had really bad seizures for 10 years. He couldn't hold a job. He, you know, couldn't function around the house. It was just really bad. And so because of this, he was pretty desperate, and his doctors were pretty desperate too. And so he allowed them to go ahead and go through this experimental neurosurgery in which parts of his hippocampus were removed. So I think, no, I don't have pictures of that. But so it was experimental, and it was really just kind of like this Hail Mary last ditch effort. And it worked. It turned out that the brain tissue in his hippocampus, that's where the seizure activity was originating from. So he didn't suffer any more seizures after they removed both right and left hippocampi, but he had permanent amnesia. So he was actually able to remember some things from his past, so maybe some childhood memories, some facts about himself or events that happened before the surgery, things about his parents, but he wasn't able to form new memories at all. So what was there before the surgery was there. Anything that happened after that, gone. So for example, if he met somebody who then left the room, within minutes, he had no recollection of them or what they talked about. So if you went and you said, oh, you know, hi, Mr. HM, how are you doing? Let me go ahead and get you a glass of water before we get started with our interview. Got him water, came back, you'd be like, hi, who are you? You'd be like, do you remember we just talked? No, I've never met you before. And so there were two researchers called William Scoville and Brenda Milner who were really intrigued by the case of HM. And through their work, they were the ones who were actually able to make this connection between the hippocampus function and memory. And so for the rest of HM's life, he was studied pretty extensively. And through this, neuroscientists and other researchers learned that the hippocampus and the surrounding areas in the brain help to transform our experiences, our perceptions, and sensations into what are then, you know, consolidated into memories that then have a lasting presence in the brain and can be, you know, retrieved and things like that. But for HM, this process of taking experiences and consolidating them into memories, that couldn't happen. He no longer had the brain structures that allowed for that. So he experienced every aspect of his daily life, like eating a meal and taking a walk, as the very first. So every time he woke up and ate breakfast, he thought, oh, this is the first time I'm eating breakfast. These eggs are delicious, even though he might have that same meal every single day. Like I said, meeting people, right, that he didn't know before the surgery. He would reintroduce himself to these researchers, Scoville and Milner, every single time he saw them, even though they might have been working with him for 10, 20 years. If you've seen the movie 50 First Dates, this is a similar, this is kind of based on HM, where, you know, every day you wake up, you don't know who this person is, what you've talked about. And so what was really interesting, though, these researchers found out was that his intellectual abilities, his personality, his perceptual abilities, they were still intact. So they still remained. It was just more about this memory making aspect of cognition that couldn't be supported anymore because those structures were gone. What was also interesting too was that, so even though his, what we call conscious or declarative memory could no longer form new memories. So if you ask them, oh, what was the name of the person you just met? He couldn't tell it to you. But his unconscious memory abilities were still there, interestingly enough. And so he actually, he was able to learn over time. So he was able to learn like tracing like these elaborate patterns that researchers had him do, even though he couldn't tell you that he remembered them. So you could ask him, hey, do you remember these patterns? And he'd say, nope, I don't know you. I don't remember these patterns. But he could, you know, you could ask him, well, why don't you just go ahead and draw something that comes to mind? You know, see if anything comes to you. And he could produce the patterns, but if he was posed with the question, produce these specific patterns, he couldn't do it. And so it also demonstrated, okay, well, memory is more complex than we've thought too. There seems to be both a conscious and unconscious sort of process to it. And so, yeah, so those are case studies, and HM is a very famous one. And like I said, he continued to be studied for the rest of his life. And cognitive psychologists also use self-report measures to study cognition. And so this can involve things like posing questions to participants or observing them engage in a task. So some examples that are really popular are surveys, clinical interviews where you're asking more open-ended kinds of questions, and also just basic observations. And I don't know if any of you have had a chance to take child development yet, but if you have, you might be aware of the fact that at Westmont, we actually have an observation room that can be used for children, and it features this, it's pretty elaborate, but it's a two-way mirror. There's all this recording equipment, and so you can have the parent and the child in the other room engaging, and you as the researcher can be observing this happen in real time. And then a last source of research has been computers. And these have really allowed us to study cognitive processes in depth, in addition to serving as one possible metaphor for how the human mind functions, like we talked about on Wednesday. So for example, we can use computer programs to write code that allows us to see an imitation of human performance on a particular task. There are also a variety of programs that can be used to more deeply analyze our performances. So for example, I know that Dr. Saad in the past has done some research on like facial expressions, emotional expression in the face, micro expressions. And I know that there's actually this really complex coding system. I think it's called FACS or something like that. Anyway, and you can use the recordings that you've taken to really break down frame by frame these sort of facial muscle patterns that correspond to the expression of certain emotions like anger or sadness and things like that. And so we're able to not only analyze, but then also generate simulations of how, based on the research that we have so far of what we know how humans think and perform, how we think that they might go about solving a problem or organizing something. Which you can imagine is a really nice workaround from things like you trying to use animals to simulate, okay, well, what would happen if we, you know, took the hippocampus out here? With a computer, we can do a lot to investigate cognitive processes outside of the lab, which is really nice too. So that is kind of the extent of the more common research methods in cognitive psychology. Obviously, all of these aren't going to be available to you as you think about designing your own lab. Unfortunately, Westmont has yet to get its own MRI machine or something like that. It only costs like a couple $1 million, so I'm not really sure what the holdup is on that. But some of these other methods are going to be a lot more accessible to you and your group members as you are designing your own studies throughout, or your own study throughout the semester. So hopefully this has kind of been some good food for thought in that particular instance, as well as just in general, as we go through talking about cognitive psychology research and findings. Hopefully this helps make more sense of how we've gotten to some of those conclusions. Before we end a little bit early, does anybody have any questions on research methods in cognitive psych? Hi, okay, I think I dropped out there. All of a sudden Zoom stopped working and then it started working. I don't know what part I was cut off at, but I just wanted to check in. Does anybody have any questions about research methods as it pertains to either generally the field of cognitive psychology, or maybe questions that have come up in your mind as you were thinking about the research that you might be doing this semester? All right, well, I'm going to take the overwhelming silence as a no. And just know if you do have questions, we can always continue to talk about them as it pertains to your research. Again, as a reminder, we don't have class on Monday. We don't have a video lecture to watch. So please enjoy that time off. Take care of yourself. So do make sure that you have your responses done for lab by Monday night. Is that right, Lydia? That's the timeline? Okay. But yeah, well, I hope all of you have a great 3-day weekend, and I look forward to seeing you all on Wednesday. All right, everyone, take care. Bye.
Thank you.
Thank you.